if you should have FIJI installed on your systems and updated. If you want to follow me with the slides and the demo that I'm doing. Also, you can follow along with the slides as we go along. You can find them at imagej.net/presentations. So the website's right here. So imagej.net/presentations with a capital P. And then we're following the living workshops that are linked up at the top. And we'll start with the introduction to FIJI. ImageJ, FIJI, same thing. We'll talk all about that today. So hopefully today we'll get through the introduction and then also segmentation workflow. So that's the goal. But we have a lot to cover, so I'm going to be moving quickly, very quickly. But this workshop is not intended for you guys to walk away feeling like, I'm a super expert at ImageJ. This is a bunch of cool stuff that ImageJ can do, and here are some tools that you can use, and exposure kind of deal, okay? So I will be going quickly. That's all. And two, we are recording this today to actually create a resource online for other people as well. So still saying that, don't be shy with asking questions. My priority is you guys here in the room. Allison is here to help as well. So if you have issues with your particular setup, like you missed a step or you just need a quick help, just raise your hand. Allison's walking around and checking to make sure everyone's doing okay. Also, if you have a question, she has a microphone. Ideally, we'd like to record your questions for the sake of this video, if that's okay. But if you don't want to be recorded, you can always just wait till the session's over at the end and ask me questions. We'll stick around a little bit at the end. Stopping the video, and then you can ask questions freely if you want. I think there's the Wi-Fi is the Discovery Guest that everyone can have access to. So these slides, also just a note that these slides are in a thing called Reveal. So if you get lost, you can just move them through them using the arrow keys or spacebar. But if you get lost, because they're actually two-dimensional, you can just hit Escape and move through the slides. See, there's like one that takes down here, so they're 2D slides. So if you ever get confused with where we are finding the right slide, just hit Escape and move through with the arrow keys. So again, the slides are at our ImageJ wiki page, so imagej.net/presentations with a capital P. If you guys just have FIJI opened on your systems, you can just follow along with what I'm clicking. But if it's too fast, then don't worry. Again, these slides are always available for you as a resource to go back to. So we always have them posted online. So you can always go back and use them as a resource. There's active links in all the slides. So you don't also have to stress about making notes. But does anybody have any questions before we get started? No. You all look very intent on setting up your systems, which is good. I like your faces right now. For those of you guys walking in, just grab a seat, pull up a chair, get comfy, and open FIJI on your systems as well if you want to follow along with the slides. The link there is at imagej.net/presentations with a capital P. And we'll start with the introduction to FIJI slides. And again, if you guys have questions or need help, just raise your hand. Either I can help you verbally. Allison is also in the back of the room. She's walking around to make sure everyone's doing okay. And I'd say let's get started, because it's 2 and we've got a lot to go over. Well, we'll wait for you guys to move the table. Okay, let's get started. So what is ImageJ? So ImageJ is an open source software package that's specifically built for scientific image analysis. Okay, this is not Photoshop. This is a tool for doing science, which is why presumably you're all here, right? So it's really been designed to be aggressively transparent, as this leads to open science, and not to mention the fact that we are we're federally funded by taxpayers, our work in building this system. So ImageJ is built on others' research, right? And it's extensible, and of course, science is the same. So just as you wouldn't publish in your paper, for example, I added 500 milligrams of chemical A. Like, you need to disclose what that chemical is, right? And nor should you treat image data in that same manner by using some black box of a software for your image analysis. So ImageJ is all about reproducibility, so much so there's even a command that bundles up the exact version of ImageJ you are using, so you can even publish a link in your next paper so others can actually reproduce your results exactly as you did. So again, it's about reproducibility, transparency, and I know I'm already hammering this into you, but it's really important and really important to us as on the ImageJ team. So obviously all of you have heard of FIJI, ImageJ, yes? Do you know the difference between the two? No? Some are saying no. Well, actually, FIJI stands for FIJI is just ImageJ. So it's pretty much the same. It is the same thing, except that FIJI is really just a collection of image analysis tools and plugins that was made specifically for life scientists. So it just comes with like a bundle of different tools and plugins for you to use immediately. So everything we discuss in these workshops today is in the context of this distribution of ImageJ is FIJI, right? So these are just like, for example, some of the tools and methods that you can carry out in FIJI. In general, opening, visualizing, multidimensional microscopy images, registration techniques, segmentation, which we'll go over in the latter part of this workshop, image measurements, 3D volume rendering, and so on. So the ImageJ Wiki, this is our go-to place to learn, right? So for beginners as well as advanced users and developers alike. There are 3 main calls to the ImageJ action. So this is like our homepage. This is where if you have questions or whatever, this is one of the first places you guys should search, is the ImageJ Wiki. And there's 3 calls to action, like I said. There's the download, so download the different versions. To learn, those are typically for beginners. If you're interested in different image analysis techniques, that's a great place to go in. And then for developers, so people who are interested more in building tools and scripting, a little bit more advanced techniques, technically, that's where you would go. So we've spent a lot of time and effort to make this wiki approachable, but it's not perfect, right? But it's a wiki, so you guys can actually help us make it better and edit it, which would be wonderful. So if you come across a page, something doesn't make sense, or even if you see a spelling error or something, you can actually edit this website, right? It's built by the community of users and developers of ImageJ. Okay. Also, if you're trying to search for ImageJ resources, you can use this targeted search portal. There's a link here at the bottom. It's search.imagej.net. It's a targeted portal for ImageJ. So you can search more specifically on the website, on our wiki page. You can search the code if you want to know specifically what an algorithm is doing and how it's implemented. You can search the ImageJ forum as well. So this is one of the big takeaways for today's workshop that I hope, there's 2 big takeaways, and this is number 1, is that the guys use the forum. So this forum is an amazing place that we created recently within the last year and a half, as like a place for the community to gather online to help each other, right? So obviously you want to save time, but of course, and more importantly, you want to do better science, right? So there's a whole open source community of ImageJ's users and developers as well as experts in image analysis that are all here on the forum that can help answer your questions. Even if you think it's super basic, do not worry. We get basic questions all the time. We get super advanced questions all the time. It's a whole host, a whole mix. It's really there as a resource to get you guys going with using ImageJ as well. Like, it's a great place to search for stuff. If you're getting started with segmentation, people have posted images of like, oh, these are these particular cells that I need to segment, and can give you kind of clues as to where you can start your own analysis. So yeah, this is a great place to go. So that's one of the big takeaways from today, is go to the forum, forum.imagej.net. What's also great about the forum and ImageJ in general is that we have a huge user community that really spans the globe. So what's cool is that sometimes if you're up late, you're thinking about your question, you post something on the forum, you could potentially wake up the next morning with your answer because of the time change. So many users across the globe are on there. You know, it's a really great tool. And this is what the strength in ImageJ is, that community that exists and that you have access to. And one of the ways you have great access to is via the forum. So the other big takeaway today, so those are two things that I want you guys to really, if you walk away with just remembering two things, it's the forum and now this, the command finder. The command finder are really going to save your life when you're working in ImageJ, and you'll see a lot of things are hidden in different path menus. So Control L is your friend. So we'll just switch over to FIJI now. And to activate the command finder, you either hit Control L or just L. And so now you can actually search for different commands. For example, I want to open a sample image called blobs. This is one particular sample image we will open later. And I want to do a Gaussian blur. So instead of having to, like, where was that function hidden in the menu, I can just type it in directly in the command finder and boom, I can run my commands. So it really just helps streamline your workflow. You don't have to like click and drag and search for stuff all the time. So command finder, shortcut is Control L or L, okay? So the main window of ImageJ, wow, isn't this impressive? This software is a beast, right? Okay, it's not really impressive looking, we know. It's very simple, but really there's a lot going on under the hood here. So it's really a powerful tool for you guys. So looks can be deceiving in this case, okay? So there's three main sections of the window. There's the menu bar, which contains things like the file, edit, image, process, analyze, the different plugins. And then there's also the tools. So actually tools are things for like drawing ROYs. So you have like squares, rectangles, circles, lines. So if you see some of these guys have a little red triangle at the bottom of that cube, that square, that means there's other options that are hidden inside. So if you actually right click on those, specifically, you'll see a little drop-down menu. So for the line, for example, the default is a straight line, but you can also draw a segmented line or a freehand line. And also those particular options that have those particular ROYs that have that red triangle, if you double-click them, you can also adjust other parameters that are hidden there. So if you double-click, you can actually adjust, for example, the line width of that you're drawing. Okay, so those are just kind of like hidden little secrets in there. Then you also have the status bar. So if you actually hold the mouse over, click on the status bar, you can actually see the version of ImageJ that you're running and the version of Java that it's using. And a lot of times if you come into problems like bugs in your software or whatever, a lot of times on the forum, if you post a question, people are going to ask you what version you're running of ImageJ as well as Java, so that's just a quick and easy way for you guys to find that information and let those developers know. Okay? Yep. So also, sometimes, you know, if you're, like for example, you're opening, I'm just going to do this as an example, you're opening tons of images, tons of images, right? And like your main window, sometimes all these windows get popped up and you can't find your main window again. All you have to do is hit enter and it'll bring it right to the front, which can be handy. Instead of spending all this time moving windows around, you can just hit enter, okay? And the main window comes right forward. Okay, so I'm just going to close all those. Okay, staying up to date. So ImageJ2 is the newest version of ImageJ. Don't worry about this ImageJ2, ImageJ, FIJI. For your purposes, they're all the same. So ImageJ2 is just the newest version of ImageJ that FIJI is using. And one of the main features it leans on is this updater. So you'll see when you start FIJI, it'll ask you if you should update. Like normally we recommend that you do, right? Because that way you get all the newest bug fixes and changes and updates to all the different plugins that are packaged within FIJI. You can also manually go to the help window and update from there. So actually we apologize, the help window is a bit of a mess at this point and it's on our to-do list. Unfortunately, there are 3 different places where you can update ImageJ on this help menu list. So the 2 that are right here at the bottom, so this update ... as well as update FIJI, actually do the same thing. So we apologize, we will fix this. But for your purposes, these bottom 2 updates will carry out the same thing. This upper one, this update ImageJ, is specifically for the legacy version, the older version of ImageJ. So that one you don't really have to worry about, okay? Just so you're aware that those exist, that's where they are. Okay. So memory management. This is just something I want to mention because it's important to keep in mind when you're working. When opening images in ImageJ, it reads in the entire image into memory for better performance, essentially. So obviously this can be a bit of an issue if you're working with large images. So in general, it's a good idea to just kind of have that in mind, which is why I'm mentioning it here. So when you first launch ImageJ, it's going to guess how much memory it should be using, which it usually estimates about 3/4 of the RAM of your system. And most often that's good enough. But sometimes you need to tune it a bit, right? So actually this is where this memory and threads comes in. So if you search in your command finder, just type in memory, you get both of these options we're going to talk about. So first, memory and threads. So you can actually increase the maximum memory that your system is using. You can change that number and then you just have to restart ImageJ for it to actually take hold. As well, you can work in parallel threads. So some plugins will use all cores. So this is just a way where you can tune the CPUs that are used at once. But usually default is fine. But this is where it exists if you want to tune it yourselves. Also, the other option, monitor memory. This actually just does a log of the memory that's actively being used on your system. So if you're running a script, doing any batch processing, and you notice that parts of it are a bit slow, you can actually have this monitor memory running. So you can see in your script maybe where things are slowing down a bit to try and optimize its performance. 2, for example, if you click the status bar, this is just kind of an interesting thing to keep in mind. It does a little memory dump. So you'll see a little blip there in the memory. So if you actually just click the status bar, it does a memory dump of everything you don't need, just kind of trashes it and clears the memory a bit. So, okay. So step 1 would be what? Obviously opening your data, opening your images to take a look at them, start your analysis. So I'm sure you're all aware that there are literally dozens and dozens of proprietary file formats for image data, right? Like Zeiss has their own file format, I don't know what other types of images you guys are using, but each one has its own file format. So ImageJ does what it can to really open them all, in a good way. But obviously, this is a problem in general. So it does its best, like I said, ImageJ, but there are 3 main ways that you can open images. The first one is just the simple drag and drop. So you literally will just have an image here, and you can drag it onto the status bar, and it will open the image. This is the same as if you go File, Image, Open, okay? But we don't actually recommend doing the drag and drop method for your data sets, specifically for TIFFs, because sometimes the metadata, it doesn't really work in every case that the metadata is read incorrectly. So I'll do it just for simple PNGs or JPEGs or whatever, but if I'm working with my own raw image data sets, I never drag and drop my images in. Primarily, I use bioformats. So have any of you heard of bioformats before? Yeah, a few of you? Okay. So basically, it's a library for opening various file formats. So it can support handling different files. So personally, like 9 out of 10 times, this is what I use. This is honestly my default option for opening images, because now you know that your metadata is being handled correctly and read in correctly. So let's go ahead and do that. So just to run bioformats, we can search in the command finder, bio-formats importer. Okay, so it's bioformats, and then start typing importer, and you should see it there. So bioformats importer, and I'm just going to open a TIFF that I have here. And this is the window that should pop up. So you've got different options for viewing your stacks, and what's great about bioformats is they have an information window that describes all the different options, kind of gives you an idea. Also, again, talking about memory management, if you have a huge image, again, you can actually open it as a virtual stack, so that's an option here. So basically, it's a read-only way of reading in your image. So if you have a huge confocal stack of images and you move to like slice 25 and you manipulate that image a bit, like you adjust brightness and contrast or whatever, and then you move to the next slice, it's going to revert to the original view. Like, so that those changes that you made aren't saved, because again, it's just a read-only view into it, but it allows you to kind of quickly see your data. You can also crop images on import if you want, if you're working with large images. So yeah, so there is a third option. I'm just going to mention it here quickly. But right now, we're not going to go into too much detail because it's not really up and running yet. But I just want you guys to know that ImageJ is actually developing our own file format reader called CIFIO, and that one day it's basically like a bioformats for ImageJ. So that will actually soon be the default open images. So now we don't have to deal with this. Should I use drag and drop? Should I use bioformats? It'll just be open image. Simple. But unfortunately today, it's not so simple, but just use bioformats importer and you'll be good. Okay? Okay. So pixel types. So do you know what I mean when I say like 8-bit data? Have a general idea? Right? So, okay. So bits basically refer to the number of possible values that you can give each individual pixel, right? So each pixel is a measurement, but what we need to do is decide on a precision for that measurement. So with more precision comes more memory, takes up more space, right? So for example, the 3 different bit types here, we have an 8-bit, which is integer values ranging from 0 to 255. So that's literally just 2 to the 8 values. Pixel values you have options for. Then 16-bit is integers values ranging from 0 to 65,536, 35. So that's 2 to the 16th different values. So that's essentially what we're discussing here. 32-bit is a bit special. It's a floating point number. So that refers to actual real numbers. But in the end, you have a bit of a trade-off with range and precision. But essentially, it's a scientific notation with limited numbers after a decimal point, and it also takes into account sign, so negative values and positive values. Yeah. So that essentially are the pixel types that ImageJ can handle. So yeah, knowing the limits of your data. So we're just going to do a quick demo here. And we're going to open actually a new image. So file, new image. And we're going to create a new image that's a 32-bit type filled with a ramp and just a small guy, just a 20 by 20 pixels. Okay? So if you want to follow along, we're just going to create a new image that's 32-bit type filled with a ramp and 20 by 20 pixels. So when we open this guy, we'll get this teeny little image here. And if you hold the mouse over the center of the image, you can actually just use the plus and minus keys on your keyboard to zoom in and out. Okay? So now, once we're zoomed in a bit, you can actually probe the different pixel values. So on the status bar at the top, you'll actually see the position of my mouse as well as the pixel value. So the value is, in this case, is 0.25. So we have values ranging from 0 all the way to 1. But now what we're going to do is just do some math on these guys. So we're going to multiply, these are really small pixel values. We're going to multiply them by a really large number. In fact, we're going to multiply them by 100 million. So that's a 1 followed by 8 zeros. Okay? So if we multiply it by 100 million, okay, something weird happened to the image, right? Don't worry. It's just a display thing, because previously we were just looking at pixel values from 0 to 1. Now we're looking at pixel values, in theory, from 0 to 100 million. So this is just like a viewing thing, a display thing. So we just need to, the first thing you should think of is like, no, nothing wrong happened with my, nothing wrong happened with my data. The only thing I need to do is adjust brightness and contrast. It's just a visualization thing. So if you just type in the command finder, brightness and contrast, so we're going to adjust that by clicking auto, and we're just back. Again, we can see our ramp. It's just adjust it after we did our maths. So now if we're actually probing the pixels, we can see there's some wonky things going on in our pixel values. Do you see there's some weird results that happened? Yeah, the numbers aren't adding up, right? So because we are increasing this precision in our 32-bit float values, we're multiplying by these small numbers by really large numbers, there's an imprecision there. Like, computers are discrete. So it's just important to remember that the maths in these cases are not always perfect, which can seem scary, but don't be scared. Okay. So it's an approximation of space, essentially, right? Okay. Oops. Okay, so getting to know your data. So obviously the first thing you want to do is just look at your data, but what's the best way of looking at your data? The best way of looking at your data is looking at those pixel values, right? So let's open this example. It's called boats. So in the command finder, again, just L or Control L, we'll type in boats and open that guy. So we can see there's boats and everything, but what we want to really do to get our science out is to look at those numbers, right? So let's use the histogram. So that is this histogram, analyze histogram. So now we're talking. Now we're actually seeing all of our pixel values of all of our pixels in one go in this histogram. And we can actually probe the histogram. If we move the mouse over it, we see, for example, on the bottom right-hand side, this gray value, which is 138, I've got 4,381 pixels in my image that equal that value, okay? So two, the histogram also gives you great insight into your acquisition settings, right? Like, did you set up your scope correctly? For example, look at this histogram. There's obvious issues here. Anybody see what the problem is? Like, you have saturated pixels, right? You basically haven't filled up the entire histogram. You've got lost information here, okay? So then that would be a sign to go back and you need to acquire new data, okay? Okay. So profile plots, so that's another great way of like looking at your data sets. So let's go back into ImageJ and open that blobs example. So it's called blobs. And what we're going to do is use that straight line ROI. So just use a straight, you could use a curvy line if you want. I'm going to use a straight. And just draw a line across any of the blobs, any number of the blobs, it doesn't really matter. And then we're going to plot the profile. Plot profile. So we're actually going to see the data across that line. The pixel values across that line. Okay? And just across that line. So again, this kind of points to really looking at the numbers in your data is the true source of information, right? So for example, do you notice anything strange between this plot and the image itself that might be a little bit backwards than What do you think? Anybody? Yeah, it's plotting gray value, because we're dealing with an 8-bit image, so we have values from 0 to 255. So if you actually look at the pixel values over the blob, the dark blob, we get pixel values that are relatively high, right? And it's only because, so for us, a high value is usually white, something that's white. Low values are usually dark. So actually, it's just showing us that this image is inverted. And actually, it tells us at the top of the image it's actually inverted. So it kind of cheated a little bit, but that's the thing. Just because the blob is dark doesn't mean the pixel values are low in any case, in this case, right? It's not until you actually probe the pixel values that you can actually see that, all right? Also, for plotting profiles, I just want to let you guys know you can also plot them in Z. So if you have confocal stacks, you can also use this tool called plot Z-axis profile, or Z-axis profile. Okay, so I just want to take a quick respite from FIJI itself and doing stuff in FIJI and just talk a little bit about some image processing principles. So looking at this image, this is a pipette, right? So what I'm asking you guys is, what does this image tell us about the volume of this pipette? And no, you don't have to speak French in order to answer that question. So we have this image. Can you tell me anything about its volume? I mean, in order to do that, we have to make some assumptions, right? I mean, obviously, this isn't even a picture of a real pipette. This is a computer-generated thing, right? But if we set a scale, so we know a length here, that could give us some idea as well. You know, we see there's shading, so we're assuming it's a 3-dimensional object. So assumptions need to be made in order to extract data from this image, right, regarding the volume that this can hold. But the data is limited in this case, right? We don't have that much information here. But that's really a huge point. When you are acquiring your data sets, that's what you have to keep in mind. At the end of the day, after all of the preparation you put in designing experiments, carrying out experiments, your biological samples, preparing them, staining them, prepping them, imaging them, choosing the right imaging modality to use, acquiring it in a good way, sampling your data correctly. At the end of the day, all you have are those sets of images that you acquired. That's it, right? So this is the data that you have to work with. So your job is really, of course, to take great care in all of those steps leading up to that, but then also do everything you can to save the best data possible. So saying that, not all image file formats are created equal, okay? So there are some, and most notably JPEG, that can actually corrupt your images. That sounds scary, and it should scare you, because that's exactly what you don't want to do in this case when you're working with your biological data sets. You want to preserve as much information as possible. So with JPEG, it's not just a matter of losing information via lossy compression. The values themselves can actually be corrupted. It alters the original data. JPEGs really was only made for human interpretation, right? Just for a quick look-see, not for scientific data sets. So when in doubt, you should always save your data sets as TIFFs or as the original file format of the vendor you're using, okay? Just to make sure you're really taking care of that metadata. So the scale, gray values, calibrations of your system, all of that will be preserved, okay? Also, pixels are not just little squares. Pixels are not just little squares. They're actually point samples. They're really Gaussian distributions from a single point source, okay? So on the left, our default in image J is to actually render pixels as little squares. But you can actually change that. I just want to tell you guys, you can actually change it if you want. In edit options appearance, you can actually interpolate around those images, and then you get this smoothed out Gaussian, but it doesn't do anything to your actual pixel values. It's just, I don't know, easier on the eyes, I guess, okay? But our eyes can be jerks, and our eyes don't always do us favors, okay? So there are a couple things that actually, if you open this sample, it's called Adelson squares. in image J. So ADEL, Adelson squares, is the sample. So if you look at these two checkerboard boxes, the ones that have these dark diamonds in the center, just looking at them with your eye, would you say that they're the same gray value? I mean, obviously I'm trying to trick you, but so are your eyes, right? It's not just me. So actually, because of the checker pattern that we see, we assume that the upper square is darker than the bottom one, despite this shadow being cast, right? The thing is, our eyes are taking into context this whole story, right? But if you actually move your mouse over those pixel values, the value is 125 on this upper square, and it's also 125 on the bottom square. But because of that story our brain creates, we see two different gray values, right? And of course, these types of things exist in our biological data sets as well. So, when in doubt, measure, 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 measure. Also, there's two other examples you can open if you want to play with them a little bit, but I'll just quick say something about them. So we have comparing lengths. So on the left side, these two red lines are actually the same exact length. And I can tell you multiple times I have opened these images to try and convince myself of that, and yes, it's true. They're the same length, but our brains interpret this as like a three-dimensional object, like a hallway. So we, again, our brains assume that the red line on the right is longer than the one on the left. And then also on the right side, this example, these are all straight lines, horizontal straight lines, totally straight. But because of that context, again, this alternating of black and white squares and the different lengths of them, we see wavy lines, right? So I know these are really manufactured examples, but again, these types of things do exist to some extent in your biological data sets, okay? If you're still not convinced, you can always go back and open these and do some measurements yourself just to see. And it's not just lines and shapes and whatnot and shades of gray, it's also color. So this is an example where I'm just gonna actually save this image on my desktop and then open it quickly in FIJI just to show you. Also another cool tool that you can use. I don't know. Okay, so I just did the quick drag and drop, which is bad. I told you not to do that. But for demonstration purposes today, I'm going to drag and drop a lot. Okay, so, but don't do that with your data sets. Okay, so what we're going to do is actually select these yellow squares, and we're going to do so with the wand tool, this wand tracing tool. And actually, I'm going to double click it, and I'm going to move the tolerance to 5. So basically what we're doing is just gathering neighboring pixels that have similar values. And actually, before I do that, I'm going to convert this quickly to just an RGB color. Okay, so I've got an RGB color image with my wand tool, and then what I'm going to do is just select the yellow guys. I just hold shift bar and click, like in the center of those squares. So on the left side and on the right side. So we were selecting all of the yellow squares that we can see for now. And what we're going to do is just clear outside, just to show you that, in fact, they're not all yellow. Again, it's a contextual thing. So if you actually probe the values again, you'll see that the RGB values are quite different, right? They're not yellow, all yellow. We have yellow and gray squares. But then that's also the wand tool, which is a helpful little guy. 2, not is it just our eyes and brains getting in on the action, it's image J itself. So if you actually open this sample, striped circles, it's called. Striped circles, I'm just zooming in and out. You'll see that image J does some wonky things in order to like render this and visualize it for you. So there are stripes actually in these images. So it kind of looks like green and yellow and purple. And as you zoom in and out, the colors change a bit from blue to green. You see the stripes, you don't see the stripes, and as you continue to shrink, some really weird rendering stuff happens, some rainbow effects. But this has nothing to do with the data itself, right? This is just image J trying to render it quickly on the fly as you're changing sizes. So don't stress. That's all I'm going to say. Okay? Okay. And finally, just something that's really important to keep in mind, I think, especially when we go to conferences, when we are presenting papers, there's a large portion of the population that deals with color blindness. Red-green colorblind is the most common. So just keep in mind, this is a lesson I had to learn myself. I work in a lot of colocalization analysis, and the default in that field, again, is red and green. And unfortunately, in image J, the color default is also red and green combinations. So just keep in mind that there are better color combinations that can be used. For example, magenta and green, red cyan, okay? And then finally, we're just touching on a few image processing principles. There's a link here at the bottom of the slide, this Basics of Quantitative Image Analysis. This is a great slideshow that has a lot of really informative information on there. So if you guys want to just take a look, it's there for you if you need. But we will go back into the world of image J and FIJI, and we're going to threshold a bit. So let's get back into image J. Sorry, I use image J, FIJI interchangeably, but now you know that's okay, because it's the same thing. So let's open blobs. So when I say thresholding, you guys have a general idea what I mean? Essentially, thresholding, it's one of the most common tasks you'll do in most cases when you're analyzing your images. So basically what you want to do is separate out objects of interest from background, for example. So like cells, not cells, foreground, background. So essentially you're setting a cutoff, okay, between these two things. So there's different ways of doing thresholding. So the simplest is just this image adjust threshold. So in Command Finder, if we type in threshold, it's threshold ... 3 dots. It's kind of an indicator that that's a built-in function in image J. Something ... So we'll do threshold ... And in this case, because this is an inverted image, right? So we don't have a dark background. We actually have a light background. So we'll just unclick that. And so you'll see that you can actually manually slide where the threshold lies. You can also make an upper bound, depending on what you're interested in. There's also a whole host of different algorithms that are applied, different filters that you can choose from. And so yeah, so how do I know which is the best to choose? And as well, if I want to be robust and I want to apply this to many different images, the best course of action would be to use an auto thresholding method, right? You don't want to if you're manually adjusting your threshold and you're doing it a lot, what are you doing, really? You're introducing bias into your results, right? So you want to try and be as robust as possible and objective as possible. So we want to use auto methods when you can. So there's all these different filters there's all these different thresholding algorithms I can use. Which one is best? Well, what's cool is that in image J, we have this thing called auto threshold. There's no dash, auto threshold. So with auto threshold, you can actually try all the different algorithms out and see, just have a general view of which one you think would work best. So in our case, we have dark objects on a light background, so we'll unclick this. And we'll just run it, and now you can see, so here's the default Huang, keeps going mean, for example. These are all the different algorithms that are applied. So you can see which one works better for your particular image. It's kind of a quick view. Okay? Okay. So, regions of interest. This is also a really common task that we're doing, that we do regularly. So what we're going to do is open this example. It's called Clown, this example image. He's incredibly creepy. I'm hoping one day the clown will disappear. So let's open that clown. And actually, we're just going to duplicate him right away. One creepy clown is just not enough. We need two. Okay? So you can actually go to Command Finder and type duplicate. Well, it's in this case, it's duplicate ... right? Or there's also a shortcut, Shift Command D is a shortcut for duplicate. I always use the shortcut Shift Command D because I do it all the time. Okay, so now we have 2 creepy clowns. Yay. So what I'm going to ask you guys to do is use this bean-shaped Roy tool that's freehand selection and draw as best you can to delineate his nose. Kind of draw a circle around his nose. So when you're using this particular Roy, you just have to click and hold, because as soon as you let go, it'll close that shape. So just try, I'm really horrible at this. So just try and circle his nose as best you can. Okay. And once you're happy with your selection, we're going to measure. We're going to analyze and measure. So we should get a results window that pops up. Now yours might not look exactly like mine. And the reason for that is because you can actually select what measurements are calculated. So just as an aside, if you go to a thing called set measurements, basically in this window you can select what you measure. So if you're only interested in area and like mean gray value, for example, you can just only calculate those 2 values. But these are the options that you have. You can also choose the number of decimal places. It's also the precision of your calculations. Okay, so the point is we measured this clown nose. So like we have this, I want this area and mean information. This is really important to me for this clown. And what I want you guys to do now is take that second clown using the same bean-shaped tool. I want you guys to draw me the exact same Roy. So when I click measure, I get the exact same numbers on this clown number 2. Okay, go. Yeah, I mean, that would be super mean. We'd be here the rest of the day, and no one would ever be able to repeat that, right? It's ridiculous. Which is why, 1, it's also not a good idea to draw things manually when it comes to your Roys, right? Because it's not reproducible at the end of the day. Okay, but let's just play a little bit. I'm going to show you guys this really cool tool called Roy Manager. So let's say we want to save this particular Roy. The shortcut to saving that is actually Command-Shift-T. No, it's not Command-Shift-T. It is just Command-T. Sorry. T. Command-T. So that should pop open your Roy Manager, and you should see your Roy. Gives a little number identifier. So it's Command-T is the shortcut. Then you should get this Roy Manager popped open. So this is basically the tool you'll use to create Roys, to save Roys, to overlay Roys on other images, because that's exactly what we're going to do right now. Because now we have our like circular outline of his nose saved. We can take that other clown, the one that doesn't have a nose circled. We can actually just toggle his show all option. And now we've overlaid the exact same Roy on our other clown. Right? So now if we click measure, we'll have the same exact numbers. Okay? So that's just a way to overlay your Roys. All right. It's okay. So once I saved my Roy, it's in my Roy Manager. All you have to do is open your other image, and you just have to toggle this show all option at the bottom, and then you can measure. Then you basically have overlaid your Roy on that other image. Okay. Oops. Okay, so that's essentially what we did. Perfect. Okay, 2D visualization. So just a couple little tools here I want to highlight that are helpful if you're working with multi-channel, multi-dimensional images. So I'm just going to close everything so we don't need to save it. We're going to open this example called mitosis. And it should be a 2-channel, yes, a 2-channel Z stack over time. So you guys can just like move through the Z stacks, changing between channels. So you'll see right now I'm in channel 1. It's actually written at the top, the number of channels, the number of Z slices, the time series number. And you'll see it's all written in red. So that's, again, the default channels for ImageJ are red and green. So the second channel now that changes to green. So now I kind of know that I moved from one channel to another. But we can change this red and green. So let's be a little bit more friendly and change the red to a magenta. So you can change the LUTs. So these lookup tables, you can just change the color because it's just a grayscale image in the end. So the color is just arbitrary, just for you to visualize as you want. So we can change it to actually magenta. So in Command Finder, you can just type magenta. Personally, I always just use this little shortcut here. It's called LUT, L-U-T. And if you click that, you get a whole drop-down menu of all the LUTs that are built in ImageJ. So you'll see magenta there, for example. But there's a whole host of other ones that you might find useful and helpful. So if you can't remember the name of the color or the LUT that you want to use, you can just go to this shortcut. Okay? And channels tool. So let's say we want to just look at one. Right now we're in a composite mode, so we can see both colors at the same time. But let's say we just want to switch back and forth from one channel to another. So then we'll use the channels tool. So a channels tool, ... So if we open that channels tool, we can actually change our options here and actually just, for example, look at one channel at a time. Turn them all off. One at a time in composite mode. You can also switch to color mode. Then it's just kind of the opposite. It'll just select one color at a time as you're moving through. Okay? Or you can look at them in grayscale individually. And then also, if you need to investigate the properties of your image, making sure the channels and Z slices and everything have read in correctly, you can check image properties. I think it's just properties. Ooh, sorry. Yeah, properties, ... So image properties. So this just lists for you the channels, the slices, the frames. Excuse me, it also has the scale information for your particular images. And let's say your images aren't scaled. Actually, you actually need to set the scale for your particular images. You can do so using this tool called set scale. Set scale, ... So then you know a fixed distance in pixels. And then you can give it a unit of length and set the scale of your image. Okay? So set scale and image properties. Okay. So for 3D tools, let's open this guy just to show you one of our built-in plugins called 3D Viewer. So we're going to open the sample called T1 Head. T1 Head is the 16-bit image. So it's actually just a slice. They're like slices all the way through this man's brain. Sequential slices. So we're going to first thing we're going to do is just convert it to an 8-bit image. So to do that, you can just go into Command Finder and type in 8-bit. For me, I just know where it is, so I always just go to image type 8-bit. Okay, and then we're going to open our 3D viewer, which is our plugin for 3D visualization. So it's called 3D Viewer. Okay, so we're going to display it as a volume, and the only thing we're going to change in the default settings is changing the resampling factor. We're going to set it to 1. Just going to be a little bit faster than when we're translating and moving it around the head. So we'll just set it back to 1. And here we go. So now we can just click on him and rotate him around. If you hit Shift and click, you'll translate him, move him around the space. I think you can also just like set him back to the center again. You can zoom in and out with scrolling. So right now we have him viewed as we have him rendered in volume, but we can also look at it as with an ortho slice. So display as, so it's an edit, display as ortho slice, for example. So there's many different ways of rendering this data. So if we just go back to volume, you can also take movies. So you can draw you can take a quick video of a 3D or a 360 rotation of your image and just record that, which is always great for lab meetings and such. For example, too, you can actually manipulate the data as well inside this guy. So let's just do something kind of fun and a little bit morbid. So let's look at the guy, get him like looking at you, and then take this circular Roy, so this oval Roy. What we're going to do is just draw a circle over his face, but like try and get it like through the middle. See, I'm not really good. Through the middle of his eye, like his pupil. Something like that. It really doesn't matter. You can draw the circle anywhere you want. And so now there's options here. If we right-click inside that Roy, so once you have your Roy drawn, you just right-click and you click Fill Selection. So essentially that's going to just delete all those pixels underneath that Roy through the 3D volume. That was a right-click on the Roy, Fill Selection. And now when you rotate, you got to go back to the little hand, at the top, so we can rotate again. We actually took a slice through his head. Kind of cool. I always like doing it over the eye, because then you can see his eye socket. Maybe I need to see a shrink or something, but I think it's really cool. So it's just another way you can like see your data, okay? Okay. So that's 3D viewer. There's also another viewer that's under development called Clear Volume. So if you're interested in 3D visualization tools in ImageJ, that's a new and upcoming, actively under development, really cool project called Clear Volume. So you guys can just check it out. There's a link on the wiki page. You can just search for it there. So registration. So what is registration? So essentially it's the process of so we're moving on from 3D, so obviously. So registration is the process of transforming different sets of data onto a single coordinate system. So basically like lining things up, essentially. So what are some of the cases where you'd want to do this? Well, for example, if you're stitching lots of images together, you're going to have areas of overlap. So you want to combine those regions. A lot of you, I'm sure, have heard of light sheet microscopy. You're taking large 3D volumes from multiple angles. And a lot of times they actually embed beads in those samples. So from each angle, they use the beads inside as their coordinate system to overlay the images. For example, when I was in my previous work, I was working on a system that had a FLIM detector as well as a detector for a confocal spinning disk. So I needed to overlay my images from those two detectors. So in that case, I used registration. So we're just going to do like a quick example, like a quick and dirty registration, but just so kind of gets the point about registration in there and show you a couple other cool tools. So we're going to open these two examples. Centipede drawing in MyVart. So in Command Finder, if you just type in centipede, you'll see them both. So just open one of each. So centipede drawing and centipede MyVart. Okay, so we have two different centipedes, two different sized images, and what we want to do is overlay them and align them. Okay? So to do that, we're just going to use a quick registration built-in tool called aligning. We're basically going to align with a Roy, right? So we're going to practice with Roys again a little bit. So we're going to draw a line Roy, a straight line Roy, from the back side of the centipede to his head. Okay? So start, whichever end you start, it is directional, these Roys. Okay? So it's important that when you're doing the registration, that if you're drawing from its backside to its head, you do so as well for the other, from its backside to its head. Okay? So we're going to just draw a Roy. This is an approximation, okay? And we're going to save it into our Roy manager again, right? So we're going to hit Command T. So Roy number 1 is saved. So we draw that and save it with Command T. And then we're going to go to our other centipede and do the same, well, same thing, not the same exact Roy, but from its bottom to its head. And again, we're going to save it. So Command T. So in the end, we should have 2 lines drawn, 1 on each of our centipedes, and they should both be saved in our Roy manager. Okay? And then what we're going to do is align them. So we're going to register them. So in the command finder, we're going to type in align. So you'll see right away it pops up, align image by line Roy. So we're taking this very literally. We're going to align our two centipedes. And I think it doesn't really matter what order, but we'll just put, because I know it works this way, we'll put the myvart on top with the drawing underneath. And we'll scale and rotate them. So those are just the different kind of morphing that's taking place to get them to overlay. So put myvart on top, then drawing, and then we'll align them. Okay, so this is our newly aligned centipede myvart, but we have a problem. He disappeared. Remember what we did to fix that last time? So one, if this happens to you, don't panic. Your instinct should be, the information's still there, I just can't see it well. So remember when we did the math on that ramp image? We just had to adjust something quickly to render it again. Brightness and contrast. So when in doubt, if the thing goes black, first thing you do is go to brightness and contrast. And we're just going to hit auto. It's a quick save, and there is our centipede. So you can see now they're aligned using that Roy. So we can actually overlay them now. They're on the same coordinate system. We've registered our centipedes. Okay. I'm just going to close all again. And segmentation. Actually, this is just a little teaser slide because we're going to go over all of this in the next hour, some steps of segmentation and how to do so in ImageJ. And then I just want you guys to keep in mind, so I don't know if not all of you are going to be here tomorrow for the scripting side. This is what we're going to go over tomorrow is scripting in ImageJ, so like being able to batch process images or like folders of images. So you can basically carry out the same protocol over and over and over again. But if you're not here tomorrow, don't worry. Just look into this thing called macros. So you can actually record the steps, like pretty much everything we did today, for example. You can record them in code, and you can create these things we call macros. So macros is just an ImageJ specific coding language. It's really simple, really basic. So even if you've never scripted before, this is a great way to start. So just, you can use the recorder, this at plugins macros record, and start looking at and using code and start scripting stuff. So even if you're not here tomorrow, this is available and you can take a look at your leisure. Okay? And then we just have a couple helpful links here at the end of the slide. Again, these slides are always available, so you can always come back to them. So again, the two big takeaways from today for this section of the talk are, one, forum. Use the forum. Go on the forum. Start reading posts on the forum. It's super helpful. It's a treasure trove of information. You've got experts in this field that are there helping people and can help you as well. And two is Command Finder. Command Finder, shortcuts, save your life, get things done quickly, streamline your workflows. Okay. Are there any questions on that part? We moved really quickly, but you guys have been great. You're actually like looking at me and nodding your heads, which I appreciate. Okay. Oh, I didn't even say that, did I? I like just got straight to it. My name's Ellen. I'm a postdoc. I'm so sorry. I did that last time I gave this workshop too. I was like so excited to get started. My name is Ellen Arena. I'm a postdoc with the Laboratory for Optical and Computational Instrumentation. Yeah, so we're basically the maintainers of ImageJ, and we're here right on campus at the University of Wisconsin-Madison. The maintainers of ImageJ, yes. So every time you download it, you've got a little piece of like Madison that you're taking with you on your computers everywhere you go. Yeah. Yeah, it's maintained here. Yeah. Of different options. So the best place to start would be to go to the ImageJ wiki for all the lists of different image processing techniques that there are a whole list of different plugins, so all the different tools that are have been developed, All of that stuff is on the wiki. So again, that's just that imagej.net homepage. So just click learn and you'll see like it'll take you right in there. Actually, we can do that now. So getting started, we just point you to a bunch of different imaging techniques and plugins. Yeah, and then the almighty amazing search function, the search.imagej.net, you can also search the website directly. So if you're say, okay, I'm interested in, I don't know, colocalization. Oh, sorry, I didn't click on the website. Then you'll get a search through specifically on the wiki and we'll get you started. Okay. Any other questions? Good. Good to start with segmentation. Let's go to segmentation. So this time, if you want to follow along with the slides, again, it's the imagej.net/presentations with a capital P. And this time, we're going to do the segmentation in FIJI. So segmentation in FIJI, just click that slides link. I give a shout out to Allison Walters, who's in the back. She is a grad student in our group. So thank you, Allison, for helping us and being here. Okay, let's go. 1 more hour, we can do this. Segmentation. We kind of hinted at it before. So how do you define segmentation? Like when I say I need to segment my cells, what does that mean? Exactly. I need to define my cells from the background. So essentially it's object identification, right? So what you're doing is partitioning a digital image into multiple segments. That's like the fancy way of saying I need defined objects in my image. But before you even like go into the steps of segmentation, there's a couple things you guys need to like sit back and think about with your particular biological samples, with your images. There are 3 common assumptions, requirements, that you really need to think about before you even start to think about a workflow or developing a workflow. So the first one is, for example, like, do you have overlapping objects? So like in this case, we've got, this is just a built-in sample. It's fluorescent cells is the sample image. It's really hard to actually delineate one given this threshold mask I created. But maybe you don't care. Maybe with your biological question, all you care about is the cell population, like the entire population, what's changing. But obviously that's going to be a big issue if you want to look at cell-to-cell variation, right? Because the problem with overlapping pixels is that you've got a single pixel that could belong to 2 different objects, right? So again, just something to think about, but it's really specific to your particular biological question, right? 2, what are the shapes of your objects? Tubes, blobs, or clouds? So I know those aren't really biologically like sound descriptors, but like when I work with my computer science colleagues, that's how they term them: tubes, blobs, clouds. So tubes for maybe us biologists would be something like a fiber, right? Like a collagen fiber, collagen network, dendrites. And in those cases, you're interested in questions like, what's the orientation of those fibers relative to each other, relative to some structure in the cell, the length of those structures, the curvature. For blobs, right, those could be anything, any circular ellipsoid type objects such as cells, nuclei, organelles, right? So you want to know their area, you want to know the mean intensity within those objects, things like that. Their relative positions to each other, or counting them. Clouds would be some sort of like more sparse signaling, so like granules. You basically have regions of varying intensity, so usually you want to look at things like the intensity of those signals, the location of that signal within another object. Okay, so just thinking about knowing the structure and shape of your objects before you even start will really affect how you develop your segmentation workflow. 2, what modality are you using? So whether you're using bright field images or EM images versus immunofluorescence, you're going to have different steps in your workflows, right? As well as how does the signal intensity look? having a good signal to noise ratio will be easier to segment your objects, right, and less pre-processing steps, okay? So there are two main approaches in ImageJ for segmentation. There's one that's tailor-made and flexible, and then there's another one that kind of leans on and takes advantage of machine learning tools, which sounds scary, but it's that's super user-friendly. And it's actually relatively easy. And you can actually combine both methods in the end. So I just want you guys to be aware of that. We're going to go over both right now. So let's just get started. Why not with the easy approach first? So ideally, this will work if you have this slide open, this exact slide, because what we're going to do is save that image and open it in FIJI. So if you have to like play Scramble now for the next few minutes, just go back to imagej.net/presentations with a capital P, go to the segmentation slides, and then just hit escape, and then move to this machine learning slide, and we're going to save this image on our desktop. Okay? So this is, so while you're doing that, I'm just going to talk because I'm good at that. So this was actually a forum post by a user. I'm just going to open it and show you because it's cool because it'll show you a little bit about the forum. So this user had a specific problem. What they had are cell aggregates inside multi-well plate, and they were having difficulties segmenting it with like classical segmentation workflows. So they couldn't, because of this, you can see for obvious reasons it's difficult, even just with your eyes. Like we can see the cell aggregates, but you can also see that there's a shadow from the micro well that could cause a problem, you know, computationally as well. There's a bit of a shadow across the image. It's darker down here than up at the corner. So luckily for this particular user, one of the first responses, well, you should use this Trainable Weka plugin. Trainable Weka. Remember that, because it's a cool plugin that we're going to use today. And again, like I said, it leans on machine learning tools. So essentially what it's doing is it's being trained. Like you as the user are training the computer with your input data, and you're basically training it with a certain classifier. It's basically teaching it what to segment. So then it could be applied to multiple images, okay? So you're using really powerful tools and classifiers of Weka, which is a collection of machine learning algorithms specifically made for data mining. So in the end, what you get is a labeled result based on the training that you do. So we're going to do it, okay? We're going to do it right now. So yeah, so let's get started. So let's go into FIJI, and I'm going to cheat again and just drag and drop this guy. Sorry, just doing the quick and dirty way. But then we're going to open the Trainable Weka plugin. So it's this guy, Trainable Weka Segmentation. That's the plugin we're going to use. And the developer of this, Ignacio, is absolutely amazing. He did a fantastic job, and he's very active on the forum. So if you have questions, because using this tool, it is really user-friendly, but it does take a little bit of effort because it's so powerful. So again, you can just zoom in and out just like you would a normal image. So this is the main view of the plugin. So we have a training window, an options window here, a panel, training panel, options panel, and then our labels. So in our particular case, we're going to just have two labels, right? All we want to identify are cells and not cells, right? Essentially cells and background, okay? So we're going to go straight to settings down here on the left-hand corner. And when we click that, we can see all our different segmentation settings. So we have a whole host of different training features that we can use. We can also change, we can also run different classifiers. So the thing is, like I said, it's an investment. Like if you really have like a more complicated, you're going to have to invest a bit of time into what do these different features mean? What would be better for my particular data set? Same as with the classifier options. But again, you can, when in doubt, you've got the wiki, and the documentation on the wiki for this plugin is fantastic. So it basically explains what all these different training features are. And you can also have the forum, so if you need to ask somebody for help, okay? So in this particular case, on this forum user, they used the default settings, and it worked pretty well, but they weren't quite happy with it. So actually one of the regular image analyst gurus responded saying, oh, you should actually click these additional training features in your particular case. If you add variance and structure features and then increase the maximum sigma to 32, that should improve your segmentation. And lo and behold, it did. But he knows a little bit more about this. So don't be intimidated by that. You don't have to know everything, right? You can always ask. So in this case, I'm going to just click those, variance and structure, and then change the sigma to 32. Now, some of you might see that they're grayed out. You see that? So actually, I didn't know that this was, I found this myself. If you go onto the wiki, in order to activate those features, you have to click an update site. So I will just quickly show you. So it's just when we did like our updates for FIJI. We have special update sites for like different toolkits, toolboxes, different groups of plugins and things, projects that are ongoing. If you update, and then here at the bottom corner you click Manage Update Sites, you get a whole host of options, of other tools that you can actually have access to that are under development. So one I'm just going to quick say this, is a quick plug for BioVoxel. It's an amazing image analysis toolkit. It's called BioVoxel. So that's a really great one if you're starting out just doing some image analysis workflows in ImageJ. Jan has created even more, has given you more options via that toolkit. But again, going back to the Trainable Weka plugin, to activate those additional features, you'd have to actually click this update site called Image Science. Okay, but all of that is written on the wiki. That's how I found out, because I didn't know. So then you just click it, and then you close the window, and then you just have to like update it, close FIJI, and open it again. So for now, don't worry about it. You can just use the default settings, and it's fine. You can actually compare your results to mine when we're training, okay? If yours isn't activated. So I will add those two, so variance, structure, and then changing the sigma to 32. Also, you can change the class names. You can also add classes. So if you need to partition 3 different things or 4, you can. You can add other classes. So we'll say cells. Class 1 is cells, because class 1, class 2 isn't really informative. So we'll call one cells and one background. Okay, so now you can see our labels change. So what we need to do now is tell the computer, well, what is a cell and what is background? And to do so, we're going to use ROYs. We're just going to draw squiggles on the image. So you can choose other ROYs you want, but the default is the hand tool. Click on your image. So for example, that's a cell. I know that that's a cell aggregate, so I'm going to add that ROY to my cell label. And I'll do another one down here. And now we also want some background, right? So just a line across the background, add it to the other guy. Oh, I drew this one, but oh, yeah, obviously that's not good, but I added it. Oh, that's a mistake. But that's okay, because I can just select those traces, so you can actually just click on the traces. And I don't like that last trace I did, right, because I've got a whole host of stuff, so I'm just going to double click it, and it deletes. So double click, deletes. So I think the minimum you need is just at least one trace in each label to start training. So let's just draw a couple, like 2 to 3 for background and the cells. And then we can start training our classifier. So now it's leaning on all those machine learning tools and is going to come up with an overlay. Okay, so what we have is a 2-color overlay. Green is background, red in this case are our cells, and we can toggle it by clicking that toggle overlay. And you can see we still have a lot of that micro well that's being selected as cells. So now we just need to train again. We just need to train some more. So now we can just draw lines. For example, I'll draw a line that kind of covers that part of the micro well and add it to the background. Do it again down here. And I'll just do a couple more cells and train again. So it's just this constant back and forth of drawing lines, redrawing, training, retraining. But just be aware too, you can overtrain. It's possible to do that, right? If you can overtrain your algorithm. You'll kind of hit a peak of performance and then it'll go back down again. So it's not perfect, but you can definitely get solid segmentation with this guy. So I'd say overall, this is pretty good, what we've got. So what you can do is what's great is that you can save this classifier now, and this particular user had hundreds of images that they needed to segment. So now we've got our classifier trained, we're good. We're acquiring all of our images with the same acquisition parameters, right? So we can apply this same image processing workflow on all of those images. So we could save that classifier and then load it and apply it to many images. And what's cool is, remember when I was telling you about macros and you can record it? All of this whole plugin is macro recordable. So you can script all of this. And if you're interested in that, go to the wiki for this particular plugin and you can learn, you can read into how to script it. So you can get a result. So you get your image, your 2-color image. But then you can also get a probability map, which I think is really cool. Essentially one for each, it's an image that has a channel for each class. And what this image is telling you, it ranges in values from 0 to 1, and 1 being more probability. It's a higher probability that it belongs to the cell class. And so for the next image, it's a higher probability of belonging to the background class. Okay, so that's just a really quick intro to our machine learning plugin, Trainable Weka. And now we're going to work through the other segmentation protocol, the more stepwise version that's a little bit more tunable in the fact that you have lots of different options at each step. And kind of we're going to quickly break down the steps of segmentation. So the first step, we're going to actually download this data. So if, again, you go to this slide, how to segment, it's the pre-processing image, or the pre-process slide. There's a link here for your image to data sets that you can download. So it's actually at workshops.imagej.net. And if you just click this zip file and download it, it's just a small folder with 5 images, example images. And if you're here tomorrow, these are the images we're going to work on for the batch processing. So it'd be great if you guys if you're going to be here tomorrow, just save it now, then you've got them. So it's at workshops.imagej.net, and then just click this zip file and save it onto your desktop. Okay, so while you're doing that, pre-processing. So pre-processing steps, there's literally hundreds of options. So what's the right one? Well, there is no right. In reality, there's no exact right segmentation workflow because all of your data is different. Your biological questions on that data are different. You basically just have to choose what's best, what you consider best, and be reproducible, right? So it's the same as when you do a wet lab experiment. You have a protocol. You create a protocol that you could hand to somebody and they can repeat it. You need to apply that same mentality to image processing and creating these workflows that you remember and save each step, which is why tomorrow will be very handy because we can just script it. Okay. So basically, the pre-processing steps, you want to correct for noise, smooth edges of your objects, or normalize the background across your images. What you want to do in this step is really increase the contrast to differentiate your objects from non-objects, right? So you have a good signal, so you have good contrast to do that segmentation, setting your threshold, essentially. Okay, so I included some links on this slide. So there's different filters that you can apply. For example, median is really good for smoothing noise Gaussian is good for background, smoothing out noise as well, but it's not so great at preserving edges because it just kind of blurs everything. If it's really important for you to look at object shape, then you want to preserve those edges, right? So then there's a filter, for example, called Kurihara, which is really good at preserving edges. But there's links to explain all those different filters here. So in FIJI, do you guys all have that data set? I should have told you sooner. I apologize. What we're going to do is open that image with bioformats, okay? Because now we want to be safe. Bioformats importer. We want to make sure all of our metadata is read in correctly. So in the bioformats importer, we want to open number 5. It's C3JW 30-minute 5. That's the one we want to just open. At least you can open any of them if you want, but that's the one that's on the slides themselves, so you can just follow along more easily. Make sure the steps that you're doing match what you see on the slides. And we'll just use the default settings. And that's fine. So personally, when I'm working with my images, I prefer looking at it in grayscale instead of the color LUT, so I'm just going to change it to gray right away. Also, when I'm working on these segmentation protocols, sometimes you make mistakes, or sometimes you do like a command, and oops, I don't want to do that anymore. Well, unfortunately, in ImageJ, not all commands are undoable. You can't click the undo button all the time. So what I always do is work on duplicates, kind of duplicate as I go. Obviously, this is not really an option if you have a giant image, but in that case, you can always work on a subset. But in this case, we can just duplicate the image as we go. So the first thing we're going to do is duplicate. So again, the shortcut for that is Shift-Command-D, or you can just type in duplicate in the command finder. So we just have a copy. So the first pre-processing step, we're just going to do one. Actually, we have a pretty good signal-to-noise ratio. We're going to try and segment these nuclei. We're just going to apply a quick Gaussian just to like smooth out the punctate staining inside the nuclei. So we're going to apply a Gaussian blur. So it's Gaussian blur ... And we're just going to use a small sigma. We're just going to use the default for now. So you actually can see the difference a little bit. It blurs it out a little bit. But even in this type of image, we wouldn't have to necessarily do that first step. But again, it depends on your data sets, okay? So then now we have good contrast. We're happy with how we've set this up. So now we can threshold, just as we had discussed before. So picking that cutoff value, right? Again, separating the foreground from background, object, non-object. So go back into ImageJ, and what we're going to do is threshold ... So image adjust threshold. And now we actually do have a dark background. Dark background. So make sure that guy's clicked, because we want our red selection. So I think the default for all of you should be red. You can also change the color too for black and white, but that's not super useful in this case since we're looking at grayscale. So we want to make sure our cells are selected, our nuclei are selected. So we have a dark background, and we're going to just use the default algorithm, which in ImageJ is actually a variation on Atsu, just so you know. Like Atsu is a really popular one and usually works for objects like nuclei and stuff really well. And we're just using the auto scale. So now what we're going to do is we're going to create our mask. So in the command finder, we're going to create mask. Boom. Because now we set our threshold, right? We just want to delineate object from non-object. So now what we did is we created a binary image, a mask. So all the pixel values now are just binary. So they're values that are 0, and I think the default in ImageJ is 255. But binary images are typically 0 and 1, for example, so 2 values, right? And again, we're going to duplicate this mask, because now we're going to play with this mask. So we set our threshold now. We have our binary image. So the only information we have regarding our objects right now is morphology information. We have information about their shape, right, their size. So there's a bunch of morphological operations you can apply to your masks, such as erode and dilate. So those respectively will reduce or grow your object by 1 pixel at a time. So if you want to like smooth out your objects, using combinations of erode and dilate will help smooth them out. If you have like little things sticking out, if you dilate or erode, they'll disappear. So it smooths out sharp edges, destroys small structures. Fill holes does exactly what it says. If you have like a donut shape, it'll just fill in the values inside. So that's really helpful. For example, if you have a membrane marker and you want to actually encapsulate your entire cytoplasm of your cell, you can fill holes and close the donuts. But if the ends of your membrane aren't actually touching, you can use those combinations of erode and dilate to get them to close, and then use fill holes, right, to cover that whole area. And then there's watershed. So that's what we're going to use today. So watershed actually is used to separate closely touching, like objects that are touching. So actually, if you look at your image in the bottom right-hand corner, you'll see there's 2 nuclei that are actually kind of molded together, right? But they should be 2 separate objects. So we're going to use watershed to actually separate them. So essentially, it works really best on circular objects, okay, because it's looking for the concavity between the 2, and will essentially draw a border between them at the minimum possible distance between the 2 objects. It's essentially what watershed is doing. So let's do that now. So in the command finder, we'll type in watershed. So this is a binary operation, a morphological operation. And now you'll see, if we zoom in, our 2 nuclei are now separated into 2 objects, right? So obviously this is important if we need to count objects, for example, or your biological question really relies on the fact that you have individual cells, right? Okay. So now we can actually identify our objects. So to do that, we're going to use this tool called Analyze Particles. So now on that mask that we've watershed, I'm going to duplicate it again because I'm just paranoid that way. And I always like to work on that duplicate. So now we're going to analyze particles. So in the command finder, type Analyze Particles. Analyze particles ... So now you'll see we're going to select, the whole idea of this is to select our objects. So we have 2 filters that we can set right away. We have a size filter, so this is in pixel squared. So right now it's in a range from 0 to infinity. So obviously if your biological question has to do with the variation of size of objects, you might not necessarily want to set up a filter right away. But let's say we've got a lot of like, in this image we don't, because we don't have a lot of like noise and stuff. But you have like little punctate background noise. You kind of want to filter that out. So you could set a minimal size threshold. In this case, we could set it to 200, for example. Then there's also circularity. So that ranges from 0 to 1, 1 being a more circular object. So again, you don't want to pre-filter really is dependent on your biological question. So you don't want to introduce bias into your results, right? And then we're going to add our objects to our manager. So essentially what we're going to do is save ROIs. This is what we're doing. We're creating ROIs around the objects in our mask, and we're saving them to the ROI manager. And we're also going to exclude on edges. So this is kind of something I always do. So for example, this particular nuclei at the top, it's cut off. We like have no information really, not true and real information about what's happening on the other side, on the outside of that image. So we don't want to count it. So anything that's touching the edges, we exclude. And then we can run that, and you'll see now we have our whole little set of ROIs that are now saved in our cells. So I also just want to quickly tell you about this, because remember I had mentioned this toolbox called BioVoxel. So Jan created, just to tell you guys that this is part of BioVoxel, he created an extended particle analyzer. So instead of just having filters for size and circularity, he actually has introduced a whole host of other filters in there that are really useful. So that's Extended Particle Analyzer as part of the BioVoxel Toolkit, the toolbox. So if you're interested, the link is there, and you can always go back to it. Okay, great. So now we have our ROIs. So the next step we can do is measure, right? So the thing is, you can move through and look at each individual ROI. You can measure individual ROIs or just a subset of ROIs, but let's say we want to measure all of them. We want to see the area of all these objects, the intensity and everything. So I've already clicked on a few of these. So in order to ensure that you measure all of them, you should always first deselect. So then you won't by accident just measure one or a subset. So deselect and then measure. And then you get your printout of results, which again you can adjust using set measurements. You can change what's calculated. But you guys see an issue here? Say I'm interested in the mean intensity inside my nuclei. There's an issue with my measurement. Yeah, I did my measurement on my binary image, right? So just like the clown nose that we did before, what we need to do but if you're just interested in area, it's fine, right? It's going to get that information. But I want mean intensity, so I need to overlay my ROIs onto my original data set, just like we did for the clown. Remember, we just toggled the show all? So if you just go back to your original data set, get that guy to the foreground, and go into your ROI manager, and you can toggle again that show all option and overlay your ROIs onto your original data set. So now I'm just going to delete these old results because we don't care about those. Those were a mistake that I accidentally did on my binary image. And now again, we want to measure. Now we really want to measure. So we're going to deselect, again, just to be sure we're not selecting individual ROIs, and then measure. And now I've got my real data set. So we did it. We segmented nuclei, guys. It's a big accomplishment. It is a big accomplishment. This is a big deal. So tomorrow what we're going to do are, how many of you guys are going to be here tomorrow as well? Yeah, awesome. So we're actually going to automate this whole, what we just did, we're going to automate it and script it. And if you can't be here tomorrow, the slides are still online at that imagej.net/presentations, and you can just go ahead and look. There's code on there. You can copy and paste. And the slides should be readable enough to follow it. But that doesn't, you guys should still come tomorrow. So we can walk through this step by step. So you can actually save your ROIs. So in this option for more, you can actually save a zip file of your ROIs. So I always do that, when I'm working with my sets, because then I always save. You have your original data set. I always save also the binary mask that I create, as well as a zip file of my ROIs, just so I can always go back, because then you can always go back. You can open that zip file directly here in the ROI manager, and then overlay them on your original data sets, just to ensure that everything's lining up.
Suppose if I want to publish that one, how do I do that? Can I save that image with this yellow border?
Yes, you can burn this into a TIFF image and save it for publication. Yeah. Yep. Any other questions? Okay. So there are other more advanced techniques, active contours, graph-based methods. We touched on machine learning in ImageJ. So just there are links there if you want to read up on other segmentation protocols. So I do have a couple extra samples that we can go through from the forum, some real-world examples that are actually just kind of nice to highlight, because we have time, actually. So let's go ahead and do that. So this was, actually, I'm going to go back into ImageJ and just close everything out so we're nice and ready to go. Okay, so we're just going to close everybody. We don't need to save anything. Okay. So this was a forum post someone posted on the forum that they needed help separating these objects. So actually we're going to if you want to go ahead and do it with me, it's great to have these slides open, because again, we're going to just save this image on the desktop and just open it in ImageJ. So in their particular case, all they wanted to do is actually count these circles. But again, they needed to do hundreds of them. So they wanted a way to do it automatically without having to sit there and count each circle. So actually, we're just going to drag and drop again quickly into ImageJ. And if you zoom in, you can see why this is actually not the easiest segmentation to do, right? Because all of these circles are actually touching. But we can actually do a really quick, streamlined version of the segmentation protocol we just did to actually count these objects, okay? So let's just do it again. This will kind of help get that segmentation kind of workflow ingrained in you. So the first thing we're going to do, so our contrast for these objects is pretty good. So we don't really have to do any pre-processing, like those Gaussian blurs or median blurs and things like that. So we're going to go straight to the next step, which would be, what? Yeah, we can duplicate. It's always a good idea. When in doubt, duplicate, for sure. But then we're going to just go straight to setting the threshold, right? So let's do that. So let's type threshold ... So that image adjust threshold. But this time now we're flipping back again. We actually do have dark objects on a light background. So you actually want to see those red circles. Okay? And again, the default, the auto threshold is really good. So we can just go ahead and create a mask, right? Just like we did last time. So the next step, now we're happy with our threshold, we're going to just create mask. And boom, now we have our binary. But obviously these are going to be hard. We need to delineate each individual circle. So remember the algorithm we applied? Watershed, exactly. We're going to separate circular objects. So first, let's just duplicate this mask. Okay, so now we have two masks, and on one of them we're going to watershed. So separate circular objects. So obviously, okay, it's not perfect in its circularity, but there's actually, if you check the forum thread, they got much more, they did different segmentation steps that actually gave you smoother circles. But for demonstration purposes today, we're just going to do like a quick nerdy segmentation, because in the end, all they cared about was counting these objects, okay? So again, I'll just duplicate, but you don't really have to at this stage, because we're just going to analyze particles. Analyze particles. So we're going to make our ROIs and get our objects. So let's just set the size pixel, you can actually do a measurement and see like roughly how large your objects are. But for now, let's just take 0 to infinity. Let's just take everything. But again, we're going to exclude on edges. So hopefully in the end, we'll just have intact circles. Though there is some wonky stuff happening at the bottom of this image, right? It's cutting some of the watershed kind of cut some of these circular objects in half. So we could actually set a size threshold to be a little bit more refined, but we'll just quick and dirty again. So we're just going to add to manager and exclude on edges. And actually the overlay, if you have the show all button and the label selected, it actually tells you how many objects there are, 75. But then again, you can always just deselect and measure. And it's fine, we're doing it on the mask now, because all they cared about was counting these objects. So we see we have 75 objects. Okay? So that's one method of how to actually count objects. I mean, it gives you more information, right? Because you can have the area of your object, the mean intensity if you overlay the ROIs on your original data set, but it also just can count objects pretty easily. And there's actually another option you can do with one of your old mask duplicates. Actually, the one that's watershed. So it's also good that we just duplicated, because I'm going to show you one other option to do. I'm just going to close the ROI manager though, and close this other guy. So if you just close that ROI manager, you don't need to save those overlays. and just open that mask that you created that has the watershed. If you don't have that one, just watershed quickly the mask that you did before. And there's another tool you can use on this mask called Find Maxima to help you count these objects. So it's Find Maxima. So we're just going to use the default here, but we're going to also exclude the edge maxima. So just like with the particle analyzer, we excluded on edges, we're going to click this here, exclude edge maxima. So actually what we're doing is we're using a different ROI selection. We're using what's called a multi-point selection. So they're actually considered like a single ROI, all these points. So what we're going to do is save them into the ROI manager as we did before with just Command T. But instead of having a ROI for each individual point, since it's a multi-point ROI, we have one. So now again, you can measure. So it just finds the maxima inside that object. So now we have 75, just as we did before. So it's another way of counting in this particular case. Okay, let's just close everything then, and I'm going to go through just a couple more examples in the time that we have left. Skeletonization. So this forum post was really cool. This scientist actually needs to measure the length of this tube. And this tube is actually the digestive tract of a termite. So again, this was an issue of this particular scientist was manually drawing them, which obviously you have the issue with reproducibility and feasibility. Like, it's ridiculous to sit there and measure by hand all these termite tubes. Plus, you're not going to be able to recreate the same exact data set each time, right? So is there a way, they posted on the forum asking if there was a more robust quantitative measure that they could, method they could use to measure the length of these tubes. So someone replied and pointed out the skeletonization tool. So actually we can walk through that right now. And I'll do it relatively quickly. So if you want to follow along with me, that's fine. I'm just going to go kind of fast for the sake of time. But if you want to follow along, it's awesome because muscle memory. It'll help you remember how we did this. So again, I'm just going to quick drag and drop. And I'm going to first just convert this to RGB color. Okay, just close the other guy. So we have an RGB color. And so what's cool about this one is that we can actually just threshold this image as well, but this time we can threshold on color. So one of the responses on the forum was to use the color thresholds tool. So if you, actually when we were typing in threshold before, you probably saw it. So we're going to use this color threshold ... So now there are actually three panels here that we can set filters on for brightness, saturation, as well as hue. So color. So what we're going to do is actually take all of the brightness and all of the saturation. So the saturation should range from 0 to 255 as well as the brightness, 0 to 255. And now we're just going to filter based on color, right? Because all the digestive tubes, presumably again, that they acquired all of the images, the same exact acquisition settings, they'll be the same color. So we're just going to try and select the area that covers the length of the tube, actually in this case without connecting the tube here. So you want to make sure there's pixels, these pixels aren't connected at the end. So I'd say that's pretty good. And when we're happy with that cutoff, we're going to select that object. And then from that selection, we're going to create a mask. So again, we're going to a binary image, okay, and duplicate it just to be safe. So now what we want to do actually for skeletonization to work properly, we really need like this whole tube to kind of be filled in smoothly, because skeletonization is going to kind of find the middle of that tube, but then it's going to throw out little, I don't know how to call them, like barbs, like little lines to areas along the width that are projecting outward or inward, that are a little bit of anomalies from the median width. So what we want to do is actually fill in these holes. So we can now use binary operators, morphological operators, such as fill holes. And you'll see this guy in action, so it's going to fill in all those internal holes. But we still have some funky ones happening here. So now I'm just going to use like a combination of erode and dilate. So just to try and grab on some of those guys. So I'm going to erode now. I dilated and erode. And then I'm just going to fill holes one more time. I'd say that's pretty good. Not bad. I'm kind of happy with that one. That's fine. So now when we're happy, we've got our digestive tube all ready to go. We're going to analyze particles. And what we want to do is just select the one large object. So in this case, we're going to set a large bottom threshold just to make sure we don't grab any of those little extraneous guys. So I set the bottom threshold at 1,000, and now I have my ROI for my tube, and now we can skeletonize. And so it'll kind of show you what it does. Again, this is a binary operation, so it's working off of the morphology of that object. And now we have our skeleton. So you see there's like a middle long line. And what that is actually the largest, shortest path, right? So it's the longest, shortest path of the length of this tube. So i.e. the length of this tube. So that's exactly what that individual needed to measure. So now we can analyze that skeleton. I don't think that's the one I want. Analyze skeleton. Skeleton. Yeah, analyze skeleton. That's what I want. So now I want to make sure that I actually calculate that value. Calculate the largest, shortest path. So now in this output, it's that red line. And it's actually printed out that length in my results table. So that was kind of the answer for that particular user, like the steps, the method they could use. Again, it's all macro recordable. Okay. So let's. No, all you need to do is find the value that has the largest value for the longest, shortest path, and that is the length of the tube in this particular case. Yeah. Okay, so I'm just going to shut everything down again. We'll just do a couple more samples, real-world samples. This is just one we're going to just quick talk about, actually. So this user posted this image saying that he or she needed to increase the detail / contrast to allow more precise body shape measurements. Specifically, this individual was interested in looking at features on this tadpole. So if you actually, I'm just going to save it and quick open it in ImageJ. It's really awesome that this person posted on the forum asking this question, because in the end, if you actually zoom in and look at this tadpole, they were interested in features on the fish, or on the tadpole. But you can tell there's some issues with the image in general, right? And not to like poo-poo this person, it's just this is the tools that they had to take images of these tadpoles. But the thing is, there's some issues here, like you've got shadows being cast. You have this, if you need to actually delineate the tadpole body, the shadows being cast is going to cause issues, as well as this blue, these blue markers. Not to mention that if you actually zoom in on the tadpole, there's really, because of the resolution of this image, you can't actually answer any questions based on these smaller features, right? You'd actually have to acquire an image at a higher resolution. So this was actually really great. So somebody actually came up with a protocol to delineate the tadpole, but unfortunately more questions couldn't be answered given this data set. But that's okay. It's not a bad thing. It's not a bad user or anything. It's not like, it was just great though that they posted this question on the forum so we could try and guide them to take better images, right? To answer the questions they needed to answer. This example, this is one of my favorite examples. So this user posted this image. Whoop, I did not open the right link. This user posted this image. So what they needed to do was segment and count eggs, okay? So first they asked for help, but it's best if you actually post the original image data set. They posted the binary image, which is fine, but then what we needed was the raw data. Okay, so they posted the raw data, and from this image, they needed to segment the eggs. So my colleague Curtis, who is a computer scientist, he is a guru when it comes to computer science, but he's not a biologist. But he got to the office early one morning, and I was sitting next to him, and he was typing away. I wasn't really looking at what he was doing. But actually, what he was doing was answering this user's post. So Curtis spent about 20 minutes, I would say, 20, 30 minutes, and he came out with this amazing script for this user to segment these eggs. And he's like, here you go, boom, run this script. You've got amazing segmentation. Look at this. In this particular image, you have 1, 2, 3, 4, 5, 6 eggs. So can any biologist in the room tell me something maybe about these eggs? They're bubbles, exactly. They're totally bubbles. So Curtis, God bless him, came up with an amazing protocol to segment bubbles, which wasn't his fault though, right? So this is just something like I like to highlight this issue, this particular example, because this is a communication thing. We're all coming from different backgrounds, from biology, from computer science, and even within biology, we aren't doing the same biology, right? So to me, as a biologist, as soon as I saw this picture, I knew they were bubbles. And I felt really bad because Curtis had spent all this time like fixing it. But in the end, the user was helped. They were able to segment the actual eggs, which was the brown thing in the center. So this is really a communication thing, right? So just try and be clear and concise and realize we all kind of speak a different language. So just try and like find some common ground when describing the question that you have on there, okay? Is that it? That's it. Oh, but what we can do is actually use this example. There's one more thing we can quick do in the last 5 minutes. Why I also highlighted this is not only the communication thing, but there's an uneven illumination on this particular image. You can see it's kind of like a darkening in the background in the center of this image. So I'm just going to show you a quick method that you can do to actually subtract background. So if you have this issue, which will help you segment objects, right? So that's kind of what the response for this particular user. They needed to even out this uneven illumination in order to better segment those eggs. The actual eggs, not the bubbles. So let's just quickly do that. So I'm just going to drag and drop again and convert this to an RGB color and close it. So what I'm going to do again, the first thing I'm going to do is duplicate it. And this is an this is a JPEG image. So there's already some lossy compression that's happened here, some corruption of the values. And there's actually a built-in function called subtract background that comes in image J. But in fact, so it applies a rolling ball radius. It applies this rolling ball across the image. But actually, we don't really recommend users use it. It's kind of this inside knowledge thing that it can do some wonky things to your data. So we actually don't advise people using it. And it's kind of highlighted here. You can see that there's like some artifacts, and they're like actually just magnified because the JPEG already introduced artifacts. So that built-in subtract background, we kind of say maybe stay away from for the time being. So instead, there's another way we can actually subtract background. So we're going to duplicate this image, and we're going to call one of the, we can actually rename the duplicate background. So it's the same thing. So we're going to kind of like apply our own filter across this image, just taking into account the background. So we're going to actually use the Gaussian blur again. But instead of that teeny tiny sigma, we're going to apply a large sigma. So that's going to kind of like smooth over all the tiny little features that are across our image and really kind of just highlight the overall changes that are happening on the image. So to do that, to really kind of just focus on the background, you want to select a sigma that's roughly a third of the size of your image. So ours is like 600 by 500 pixels. So I'm going to set a sigma. You can set it anywhere between 100 and 150. It's approximate. So again, roughly a third. So I'll set 150. So you'll see we smoothed out our image, right, using this large sigma. And now we're just going to do some basic math on our image. Okay? So we're going to use this thing called image calculator. Image calculator. And we're literally just going to subtract our background image now from our original image. But since we're doing the subtraction, little warning bells should go off in your head. That means that we could potentially get negative values, right? So in order to not clip our data, in order to save the sign of those values, we need to actually create a 32-bit float result. Otherwise, any negative value is going to get clipped off at 0, right? So we're going to lose information. So then when we do this, now we kind of have like a smoothed out image with that uneven background illumination problem kind of fixed, which will help us with our segmentation steps, preceding steps. Okay, so that's pretty much everything I wanted to go over today, and we did so very quickly. So if there's any other questions, feel free to stick around for a little bit after, but like in general, like this presentation is finished for the video. So thank you very much for coming today, but again, I'll stick around for a couple minutes to answer any questions you guys might have. So thank you.